Thanks everyone for joining this month's bioCADDIE webinar. Today our presentation is by Dr. Harismendy, who is an assistant professor of medicine at University of California, San Diego in the Division of Biomedical Informatics. He leads the Oncogenomics Lab at the Moores Cancer Center, and his main interest is in the development of genomics assays and computational analysis for personalized cancer care. In the past 15 years, Dr. Harismendy's research has contributed to the understanding of transcriptional regulation and the role of regulatory variation in human diseases. You can go ahead and start, Olivier.
All right. Well, thank you everyone for listening and coming here. This project I would like to talk to you about is related to the bioCADDIE initiative and how to enrich as much as possible the data set that we indexed through bioCADDIE. And one of the elements that we decided to focus on for this enrichment in terms of metadata is the genetic ancestry of the participant in some of the genetic data sets. So this is the rationale. I mean, a lot of the human subject cohorts that are being collected in biomedical research lack diversity in terms of racial or ethnical diversity. And most of the metadata associated with these cohorts regarding ancestry rely on the self-reported race and ethnicity whenever it is available. However, molecular data that are being generated as part of the work that these studies are made for, molecular data such as SNP genotypes or genome sequence or exome sequence, even transcriptome sequence to some extent, can be used to determine the ancestry at the molecular level and even further quantify the level of admixture. So the goal of this initiative here was to include a diversity score that we would calculate for multiple cohorts indexed by bioCADDIE and the DataMed search index. And there's quite some utility, as you can imagine, to have this type of metadata and more accurate metadata in the system. First, the main objective is to facilitate the cohort selection for secondary research using this public data set. Another objective would be also to monitor the current diversity of all publicly available data sets and maybe create an incentive to assemble a more diverse cohort of ancestry that are underrepresented. And as a corollary, it would really identify, help identify underserved study areas and population that need to be improved. So as a matter of introduction, I can remind people of a number of studies that have demonstrated that our biomedical data and genetic cohort in general lack crucially diversity. This study was in 2010, and 2010 was still recent in terms of genome-wide association studies, for example, but they did a thorough job trying to understand the main ancestry that was used in a number of GWAS, published GWAS study. And their conclusion is what 75% of them were performed in entirely European ancestry and no other ancestry. So that's really a caveat of those studies. And there's actually a critical importance, even beyond understanding the role of common variants in GWAS. There's even rare variants that where the ancestry context is critical. This is an example published in 2016 by Man Rai et al. That look at hypercardiac myopathy as being misdiagnosed in African American. And here is a number. Here are different variants that are known or been published at least to be associated with these familial HCM. And as you can see, their allelic frequency in the African American is really much higher than in the Caucasian. So even if you consider all the people who have cardiomyopathy in the African American, I mean the penetrance in these African American would be less than 1%. So probably those variants in this particular African American context are not deleterious. And this is really the origin of the data that determine the pathogenicity of these variants. As you can see, the initial studies, for example, for these TNN2 variant was performed in a Spanish ancestry, or another one was performed in a Japan ancestry cohort. And interestingly, the author bring up that even inclusion of a small number of Black and American in the control cohort of those association studies would have prevented really these misclassification of those variants being pathogenic. Another example that was published even more recently in 2017 is in regard to the chemotherapy toxicity. There's a number of SNPs that have been associated with the toxicity to various chemotherapy used in cancer treatment. And as you can see in this table, a lot of these studies, initial studies, were only performed in a given ancestry, whether it was East Asian or European, and very few were actually diverse. And in the columns on the right, you noticed, for example, that some of these variants that were highlighted are highly, highly ancestry specific. For example, this docetaxel variant is absolutely not present in the Chinese, not present in African descent, and present at a minority frequency of 7% in Caucasian. And so these variants are unlikely to be relevant in context other than the European context. And finally, today we have a good way to map ancestry thanks to the effort of the 1,000 Genome Project. So the 1,000 Genome Project collected actually more than 2,500 genomes from, I believe it was 26 different populations that they located across the world, and then they grouped into these 5 superpopulations. In green would be the East Asian, in purple the South Asian, in yellow would be the African or African American, in blue the European or Caucasian, and in red the Native Americans or American ancestry. And in the next slide, we provide more of the details and the origin of these populations, and then the name of those superpopulations. So altogether, this data was assembled in a large reference panel of 81 million variants that can be used to compare any given sample to these 5 superpopulations and provide a level of admixture. And admixture is actually really critical for some population. Here is an example from a publication in 2015 where these 2 panels represent 2 different genomes from people from Colombia. As you can see on the person on the left-hand side, there is a well-balanced admixture in the pie chart between African component, Native American, and European component. And you can even further dig down, and if you have enough resolution with the SNPs, you can further dig down and maybe phase those variants and determine at the chromosomal level what's the more likely ancestry of each chromosomal segment. In contrast, for the individual on the right-hand side, you can see that the African component is much smaller. And so it becomes really important if you want to group those 2 individuals in one genetic study to understand at a given location in the genome what is the actual ancestry of that particular SNP. And this was actually performed by Laura Feyerman in 2012. She was interested in looking at breast cancer risk. And the first question she was asking is whether the local ancestry was associated with breast cancer risks. And so she identified this way a region on chromosome 6, which is here on the left-hand side, that has a very strong association with ER-positive breast cancer in ancestry of Hispanic population. In contrast, the chromosome 11 didn't show as much association. And she actually went further, and in 2014, identified the actual SNP that was responsible for this association. It's a SNP located at a cytoplan 6q25. And in fact, this particular SNP is an American, Native American ancestry or Indian American ancestral SNP that has a protective effect on breast cancer. And as you can see in the different statistical model for association, you can see a very small odds ratio for the people when considered either the global ancestry or the local ancestry or the SNP itself. So this few introduction was really to highlight the critical importance of knowing diversity of the cohort and admixture of the cohort in order to perform more accurate genetic association. So I will first walk you through some of the methods that we've used in our project, and then present you 3 vignettes on how we think we can really scale this up and how accurate our findings were. So the first is the method. As you may know, different cohorts indexed by bioCADDIE have different data type. We have a majority of cohorts that have only genotypes, but we have other cohorts that have whole exome sequencing, whole genome sequencing, or RNA sequencing. These are the 4 data type we considered for this project in order to be realistic, in order to call ancestry. Now, not all of these cohorts were eligible to perform this type of ancestry study. So we selected some cohorts that were eligible for general research use and where there were no explicit barriers preventing us from calling ancestry on these participants. And finally, we focused on cohorts that were maybe a little easier to access to when they're not too fragmented between different institutions. And so this is a list of what we started from, and this is just illustrating the size of a number of these cohorts present in dbGaP, as well as the variability in the data type they have. As you can see in the top, we have the Cancer Genome Atlas. Actually, the first 2 are related to the Cancer Genome Atlas. And the great thing about this cohort is that they have all 4 data types available, both genotyping array, exome, genome, and RNA sequencing. So this will be a great cohort to prove one of the points that we're really interested in, which is no matter what the data type of your cohort, we will always find a way, an accurate way to measure its ancestry that does not depend on the data type. And following down, you can see that different cohorts, again, have different data type. So in terms of methodology, we wanted to determine admixture and not necessarily perform what you may have seen, the principal component analysis, meaning we have a reference population, the one from the 1,000 Genome Project. So we decided to use this tool published by one of our colleagues called iAdmixed that will compare and calculate the maximum likelihood estimate of each of the 5 reference superpopulation for any given individual. So this slide has a little bit of math to explain what's behind it. So if you consider that a single individual it will have allelic, so every given SNP has an allelic frequency in the population. Those are Q. The A's are the admixture of this proportion in the different population. For example, in our case, A is a 5-dimensional vector. And so you can first calculate this weight allelic frequency at a given SNP given the admixture proportion. So that's the term Fi. And then when you measure a genotype in a given individual, the probability of observing a given genotype is therefore a function of the admixture proportion and the overall allelic frequency in the population. That's the second term. And finally, you can put together this log likelihood given the admixture proportion, the log likelihood of observed genotype G for an individual. So the goal is really to determine the vector A that maximizes this log likelihood of proportion. And so this has been implemented in iAdmix, and that's what we've used. So the first vignette is really looking at the entire TCGA data set. And so this is the self-reported ancestry in the TCGA data set. As you can see, and as we knew before, most of the samples are white or self-reported as white, and with additional ethnicity of non-Hispanic or not Latino. We have a few samples that are not reported. We have few Black and few Asian American samples. So we've used the Affymetrix SNP6 chip to call molecular ancestry and admixture. Once we call admixture on these 5 superpopulations, we define a number of additional elements. For example, we wanted to define the dominant ancestry as the ancestry that has a fraction of admixture greater than a certain threshold, and in most cases we set it to 80%. And the corollary of that is people who do not have any of these dominant ancestry are put in a 6th bin of admixed. So in the end, every single subject in the cohort can be assigned to one of those 6 categories, either European, African, East Asian, South Asian, American, or the last one is admixed. And so now how well do we do? How well does this molecular ancestry match the self-reported ancestry? So following this idea of a dominant ancestry, we can call dominant at several thresholds. And as you can see from this, and this analysis was only performed using self-reported white, Black, and Asian. And as you can see, the matching rate decreases as you become more stringent to call a dominant ancestry. But at a threshold of about 80%, you still do a fairly decent job, and 75% of individual reported, self-reported match their dominant ancestry at this threshold. Here is another way to look at it. When you select the different race, self-reported race or ancestry, either Asian, white, Black, or Hispanic, you can see that the dominant ancestry matches the self-reported ancestry. In the case of the Hispanic, we notice that the vast majority of the people self-reported as Hispanic, not the vast majority, but yeah, the higher proportion is qualified as admixed at this dominant threshold. Now, if we focus a little more on those admixed individuals, again using this 80% threshold, we notice that most of them self-report as white, 75% of them self-report as white. And when you look at their ethnicity, the vast majority do not report themselves as Hispanic. They actually report themselves as not Hispanic or Latino. And finally, once in the chart at the bottom, you notice that a large fraction of these people who have an American or Native American dominant ancestry are self-reporting themselves as Hispanic. So admixed people tend to report themselves as white and not Hispanic, the vast majority. I mean, this is a very high-level view, of course. Now, we can dig into the detail using this type of ternary plot, where we can start in this three coordinate system the amount of ancestry between two major ancestry and then the rest. So, for example, people who are self-reported Asian indeed have a large fraction of either East Asian or South Asian ancestry, and all cluster in that corner of the plot. And similarly, the people who are self-reporting themselves as black cluster somewhere between the European and the African ancestry, with little with any other component. There's a few, but very little overall with any of the other component. And that's understandable because the vast majority of these self-reported black people are African American and known to be admixed. Another example is looking at the largest share of these data, which is the people self-reporting themselves as white. And as we can see, there's also a certain level of admixture in this particular population. And finally, the people self-reporting themselves as Hispanic or Latino there's a fair number of them that are on the American and European axis, but you can see that there's a fair number of them that are really admixed between more than two different populations. There's even some people who can report themselves as black or African American, where in fact these particular individuals tend to be dominantly of a Native American background. So this was a very general overview of 10,000 or almost 11,000 individuals in the TCGA cohort. Now, that was using the SNP array, which is really the gold standard to perform this type of analysis. We know that the number of cohorts does not have genotyping array. They mostly have exomes, and we wanted to first know whether we would be as good at calling ancestry using the whole exome data. So in order to do that, we selected a subset of the individual in the cohort. We selected 100 cases that we know are coming from a very diverse. We selected them to enriched for the more minor ancestry. So we selected 25 individuals self-reported as white, 30 individuals self-reported as black, 20 individuals as American, Indian. So to have like a nice sample of the different diversity. And then we downloaded the raw reads, or the aligned reads at least, and called the variants using the FreeBias pipeline that was developed by Gabriel Marth. And we filtered for SNP that were known to belong to the database SNP and that have a high quality coverage. These are the whole exome, the number of variants in each of these self-reported population. This is what's expected. We observe about 20,000 variants per individual. We notice that African American have a higher number of variants. This is known. It's just more diversity in this population. And the first look we can have is the cumulative ancestry distribution that we get from the array and from the whole exome sequencing. So again, this is not the proportion of individual, but this is just summing up the fraction of European ancestry we see throughout the cohort, the fraction of African American ancestry we see in the cohort. And as you can tell, we do a fairly good job. The estimate from only these 20,000 variants from the whole exome is fairly representative from what we can see with a more thorough method like the genotyping Now, if we start to look at the detail of each individual, we see a little bit more noise. Here I plot in these different scatter plots in the x-axis the level of admixture observed in the whole exome sequencing versus on the y-axis the level of admixture that we measured from the genotyping array. I mean, most of these individuals are in the corners or distributing along the first diagonal, indicating that there's a fairly good correlation between the two methods, but we notice that there's also a certain level of noise. Now, one way we decided to measure this noise is, again, using our dominant ancestry classification, and depending where we put the threshold for a dominant ancestry, we make an increasing number of errors. So as you increase, for example, your definition of dominance, and you set it, for example, at 80%, you make an error in the classification between the array and the exome of about 30% of the subject. Of course, as you move this threshold up, you can see that the level, the fraction of the individual that are classified as admixed increases as well. But overall, we think that although there's some variability at the individual level, we feel confident that at least at the cohort level, we get a very good representation of the different races and ethnicity present in a given cohort. So now, how can we use this information in order to provide a summary number for people to query these data set as part of the bioCADDIE and the DataMed index? Well, I presented before this cumulative fraction of the disease, so that's a pie chart. I'm not a big fan of pie charts in general, but I have to admit it represents the cumulative fraction of European ancestry, African ancestry that we observe in a given cohort. Again, it's not a fraction of individual, it's a fraction of, chromosomes, if you want. As you can see, if you separate the TCGA by disease, it's pretty boring. They're all very European in a way, without surprises. The breast cancer is known to be one of the ones that is enriched for African ancestry, and we can see that from that particular distribution. Now, if you break down the TCGA cohort by self-reported, we can see a little more colors. Again, the cumulative distribution of the people self-reported as Asian is consistent with the vast majority of the chromosomes being from the Eastern Asian ancestry, very few from the South Asian. And similarly, on the bottom right, we can see that people who self-report as Hispanic, when you accumulate all their chromosomes together, you can see that it's a fair, even distribution between European and American, Native American ancestry with a few of the others. So we feel that this cumulative distribution captures a large fraction of this diversity for each of these cohort or subset of samples. So it's likely a very good way to represent this. Now we could take the trick of assigning a particular dominant ancestry. That means we need to make a decision on what threshold we do. And when we do that, we create this other admixed cohort. And we observe similar patterns that when you break down the cohort by disease, we don't see many differences, still dominated by the European component. If you break down, again, the cohort by self-reported, either race or ethnicity, we again see more colors, but these admixed fractions tend to occupy most of the space in these cases, especially when people self-report themselves as Hispanic. I would like to propose a third way to look at diversity, which would be a one number to summarize it all, and this number will be derived from the Shannon entropy. And as you can imagine, a type of a cohort that would be distributed between four categories. If those four categories are evenly represented, you can have a very low diversity and a very high entropy. versus if one of the categories is overrepresented, you would have a very low entropy and very poor diversity. So we calculated this entropy for the different cohort when we have individuals that are assigned to one of those 6 categories. As you can see again from the disease breakdown perspective, all those sub-cohorts are, I would say about average. It looks high. I mean, the entropy should be between 0 and 1. It looks high, but it's not that high. The lung being the least diverse one. And when you break it down by self-reported ancestry or ethnicity, we can start seeing, for example, the Hispanic now is becoming the more diverse cohort with an entropy the more diverse with more than different ancestry represented here with an entropy of 89. So now here's a proposal for those of you who are familiar with the DataMed search index. Here I searched for all the GWAS studies in cancer. I selected the one that were present in dB-GAP. It tells me we have 364 of them. And there's a number of additional metadata that I can use to filter, for example, the specific cancer type or the study design, for example. What we would like to propose maybe is to have another category that would be diversity, and with maybe 2 different metrics that we can sort this cohort based on. This minimum diversity as represented by the entropy, going from a very diverse cohort on this side to maybe a very unique ancestry cohort on that side. And finally, in complement to this diversity index, it's important to be able to sort for a specific main ancestry. So based on the 5 continental ancestry, we can request that the cohort has at least 50% African chromosomes in them, and that would be represented by this pie chart that would be displayed here. So we think it would be an excellent tool to be able to query the diverse cohort and represent much more accurately than self-reported ancestry does. So as a conclusion, I hope I explained very why global and local ancestry are critical covariates in a number of genetic studies, and that in a lot of cases, self-reported ancestry is not a reliable metadata, especially when people have a high level of admixture between 2 or 3 superpopulations. And finally, which demonstrated that exomes and SNP array lead to fairly consistent estimates of ancestry at the cohort level. And we propose concise ways to summarize cohort diversity using both an entropy measurement as well as the major or the dominant ancestry in a given cohort. So this is the summary, and I would like to thank my colleague, Ji-Hoon Kim and Max Su, and as well as Lucila Ono-Machado for the support and the entire bioCADDIE team and funding. And I'm happy to take questions.
The conference has been unmuted.
All right, thank you for the webinar, Olivier. That was really wonderful. So if nobody has any questions, we can wrap up here for today. And our next and last bioCADDIE webinar will be a presentation by Lucila Ono-Machado, where she'll talk about everything that bioCADDIE has accomplished in the last 3 years of the grant, and that will be on August 10, 2017. So hope to see everyone then. Thank you.